has been institutional coordinator of the CSIC, and she has described many different effects of thyroid hormones in cancer, in fibrosis, and sepsis. Anna, she has been awarded for many international and national awards, and most importantly, she is a key reference in research, and it's a pleasure to have her here with us today. So, Anna, you can start when you're ready.
Thank you very much, Esteban, for inviting me to the seminar and also for the nice presentation. And I'm sorry I cannot be there today, but well, next time. Okay, so I'm going to talk about the tumor suppressor actions of the thyroid hormone receptors. The most active thyroid hormone is triiodothyronine or T3, and this hormone acts in cell by binding to nuclear receptor. There are two different genes, TR-alpha and TR-beta, and these receptors belong to the superfamily of nuclear receptors that act as ligand-dependent transcription factors. These receptors normally bind to the so-called thyroid hormone response element. There are short regulatory sequences in regulatory regions of the target gene. Normally binds as heterodimer within a nuclear receptor, the retinoid X receptor or RXR, and these receptors can act either as transcriptional stimulators or as transcriptional inhibitors, depending on the interaction with co-repressor and co-activator complexes. Normally, in the absence of ligand, the receptors are bound to co-repressors, such as, for instance, NCOR, that belongs to complexes with histone deacetylase activity that will produce chromatin compactation and transcriptional repression. However, normally in the presence of a ligand, the receptor can bind to different co-activator complexes, and some of them contain histone acetyltransferases that will prepare chromatin for transcriptional stimulation. As you know, the thyroid hormones and thyroid hormone receptors are key regulators of metabolism, growth, and development, but there is also evidence that they could play a role in cell proliferation and cancer. And it's very interesting that the thyroid hormone receptors are required for proliferation of normal cells in several tissues, but however, there is also evidence that these receptors can act as tumor suppressors. And the first hint that the thyroid hormone receptors could have a role in cancer was the finding that the TR, and particularly TR-alpha, is the proto-oncogene for a viral oncogene that acts as a dominant negative of the endogenous thyroid hormone receptors. This oncogene VRB induces leukemia in birds, and also when expressed, for instance, in mice, induces formation of hepatocarcinoma. There are also alterations of the thyroid hormone receptor genes that are rather common in human tumors, including breast and liver tumors that I will talk today about. And these alterations normally lead to loss of receptor expression or activity. Also in experimental models, knocking mice that express a dominant negative TR-beta spontaneously develop metastatic thyroid and pituitary tumors, and these animals are also more prone to develop mammary tumors. Also, TR-beta downregulation appears to be associated with hepatocarcinoma onset and progression. And last but not least, thyroid hormone receptors are ubiquitously expressed in mammalian tissues, but however, cancer cell line express normally very low TR levels, and it's difficult to find a model to analyze the thyroid hormone action in culture cells. In our laboratory for almost 10 years now, we have been working on the role of specifically thyroid hormone receptor beta as an inhibitor of cellular transformation and cancer. And one of the things that we have been doing was to analyze the interaction between the RAS oncogene, that as you know is one of the oncogenes that is mutated in many human tumors, and TR-beta signaling. And we could demonstrate that the thyroid hormone receptor beta was able to inhibit transformation by RAS in these experiments of formation of transformation foci in NIH3 fibroblasts, in expression of oncogenic RAS induces the formation of a large number of transformation foci, but when TR-beta is expressed together with RAS and the cells are incubated with the hormone, with T3, there is a strong inhibition in the number of transformation foci. And when we look at the receptor domains that could be involved in this anti-transforming activity of the receptor, what we could observe is that surprisingly, mutations in this particular residue that abolish the recruitment of activators did not abolish the ability of the receptor to mediate T3-dependent inhibition of transformation. But however, we could observe that mutations in these domains that is required for the requirement of co-repressor also abolish the T3-dependent inhibition of transformation. And when we knock down NCOR in the cells, the co-repressor NCOR, we could observe that depletion of NCOR increases strongly the number of transformation foci, indicating that this co-repressor represses transformation by the RAS oncogene. And also, very interestingly, this strong reduction by T3 was almost totally reversed in the absence of NCOR. And the same happened when we look at the antitumorigenic effects of TR-beta. We conducted xenograft assays with fibroblasts expressing RAS, and as expected, they might develop big tumors after some weeks. But however, when the cells express RAS together with TR-beta, the tumor formation was abolished, as you could see here. And also, interestingly, this mutant in the co-activator domain was still able to repress tumor formation by RAS, but the mutant in the co-repressor domain was unable to inhibit tumor formation, then indicating that co-repressors are very important in the not only in the transforming but also on the tumorigenic effects of the receptor. We wanted to analyze the possible role of endogenous receptor and RAS-dependent mutants, and for this purpose, we use this model that probably you all know, that the model of chemical skin carcinogenesis. In this protocol, the animals are given first a carcinogen, and then they are treated for several months, twice a week, with a tumor promoter TPA. And this leads to mutational RAS and to the appearance of tumors, the back of the animals. And these skin tumors normally are typical benign papillomas, but some of them can progress to carcinomas. And when we look at the expression of TR-beta, we could observe a large number of cells that are positive for this receptor in normal skin and after short treatment with TPA. But however, this expression is much lower in papillomas and is almost totally lost in the carcinomas, then suggesting that TR-beta could have a suppressor role in the progression of these tumors. And to test this, we conducted these protocols in wild-type animals and in knockout mice, like in the thyroid hormone receptors. And surprisingly, we could observe that these animals, they develop less tumors. And in other studies, we could define it that this formation, the reduced formation of tumors, probably due to the fact that the thyroid hormone receptors are required for normal proliferation of keratinocytes, and also they are required for the mobilization of the skin stem cells that are necessary for the initiation of these tumors. But however, interestingly, when we look at the tumor growth along the weeks, you can see that the tumors in the form in the wild-type mice, they grow very slowly. The volume is not very big, but however, in the knockout mice, since approximately week 20, these tumors started to grow very rapidly. And both histological and magnetic resonance studies demonstrated that, as expected, more than 80% of the tumors that are formed in the wild-type mice, as expected, were typical papillomas. plus. However, in the absence of the thyroid hormone receptor, these tumors, more than 40% were already carcinomas, as you could observe here, then demonstrated that indeed the thyroid hormone receptor can have an inhibitory role in the progression of these RAS-dependent tumors. Another mechanism that could be involved in the anti-transforming effects of TR-beta is the induction of cellular senescence. That, as you know, is one, like the first barrier that a normal cell has against transformation and tumor progression. And as you know, the cells, normal cells can undergo either replicative senescence or premature senescence as a consequence of either oxidative stress or paradoxically of oncogenic stress. And this is associated with a persistent DNA damage response signaling with formation of double DNA strand breaks. And in mouse embryonic fibroblasts, we could observe that incubation with either T3 or with GC1, that is a synthetic analog that specifically binds to TR-beta, induced important reduction in the accumulating population building levels in the cells. And this was accompanied by an increase in the number of senescent cells measured with beta-positive beta-galactosidase activity. And this increase in cellular senescence was also accompanied by an increase in the formation of DNA damage foci that were identified by the co-expression of CT3BP1 and the phosphorylated form of the histone H2AX that are markers of DNA, double DNA strand breaks. And treatment with T3 or GC1 not only increased the percentage of cells that have DNA damaged foci, but also the number of foci per nuclei. And this effect of T3 appears to be a consequence of increased mitochondrial production of ROS. And as you can see here, incubation with T3 induces ROS formation. When we analyze the mechanisms, this is due to the induction of a mitochondrial transcription factor that in turn increase the transcription of many genes of the respiratory chain. And this increase of mitochondrial ROS induces the formation of foci that contains oxidized DNA labeled with the 8-hydroxy-deoxyguanidine. As you can see that this oxidized DNA co-localized with the double DNA strand breaks, and T3 increase very strongly the number of these foci. To analyze whether or not this thyroid hormone induction of cellular senescence could also occur in vivo, we used young mice, 2-month-old mice, that were treated with the thyroid hormone T3 and the precursor thyroxine in the drinking water for 2 weeks. And we applied this model to wild-type mice, to TR-alpha knockout mice, and to TR-beta knockout mice. And when we look at the liver, that is a very important thyroid hormone and thyroid hormone receptor target tissue, we could observe that in these young animals, the treatment with thyroid hormones induced the appearance of a very important number of senescent cells, both in wild-type animals and TR-alpha knockout mice, but not in the TR-beta knockout mice, then demonstrating that also in vivo TR-beta is the essential receptor isoform mediating this effect. And this was accompanied also of increased DNA damage, also in wild-type animals and TR-alpha knockout mice, but not in the TR-beta knockout mice. Therefore, we believe that, as I told you before, it could be an important component by which the thyroid hormone receptor beta could act and have actions as tumor suppressor, but inducing this tumor suppressor mechanism. In next step, we wanted to analyze the effect of this thyroid hormone receptor beta in human cancer cells. But since, as I told you before, there are not many models, no models really to study, for instance, the effect on liver cancer. We use this cell line, SNK1 cells, and we re-express the thyroid hormone receptor beta in a stable manner, and we conduct a xenograft analysis. We could observe that cells that express the receptor produce tumors with longer latency, and also the tumor volume was much smaller when the cells express TR beta than the tumor formed by parental cells. And also very interesting, the morphology of these tumors was very different, because the tumor formed by the parental cells are very infiltrative. They infiltrate, most of them, muscles, vessels, and the skin. But however, TR beta expressing tumors were much more compact, and they were kind of surrounded by this type of silver capsule, and infiltration of muscle, vessels, and skin was very strongly reduced. And also the receptor had an important anti-metastatic effect in these experiments of formation of fundamental metastases, in which the cells are injected into the tail vein of the immunodeficient mice, and we look at the formation of metastases in the lungs after 1 month. We could observe that most of the animals that are injected with the parental cells develop metastatic lesions in the lungs, but less than 30% of the animals injected with TR beta expressing cells had metastases. And also, these lesions were different because in the wild-type animals, the lesions were large and nodular metastases, but however, the TR beta expressing cell, what they gave origin is to these micrometastases with the pattern of interstitial cells. And also, the number of lesions per lung was very strongly reduced by the thyroid hormone receptor. We have been analyzing which are the genes and mechanisms that could be involved both in the inhibition of the tumor cell proliferation and the inhibition of formation of metastases. One of the mechanisms that appear to be involved in the inhibition of cell proliferation by the thyroid hormone receptor beta is the induction of microRNAs with the tumor suppressor activity, and more specifically, these two microRNAs 503 and 424 of the microRNA 16 family that act as inhibitor of proliferation were induced by the thyroid hormone, we will see. And these microRNAs, they really repress cell cycle progression, because many of the transcripts that are targeted by this microRNA are stimulators of cell cycle progression. Specifically, the microRNA 503 had been described as a master regulator of the cell cycle. When we incubated the cells that express TR beta with the hormone, you can see that these microRNAs are induced, and also the primary transcript, the pre-microRNAs 424 and 503 were similarly induced, and this is not surprising because these two microRNAs are transcribed as a bicistronic message. They share the same promoter, and in luciferase transcription assays, we could observe that the promoter of these microRNAs contain sequences that are able to mediate degree-dependent stimulation of transcription. And in fact, the promoter of these microRNAs contains thyroid hormone response elements. You can see here, sorry, in chromatin immunoprecipitation assays that the receptor binds constitutively to the promoter. In the absence of ligand, the co-repressor in core is strongly bound, but is released from the promoter in the cells incubated with T3. And in contrast, the CBP, SLC1 are perceived that are coactivators with histone acetyltransferase activity are recruited, and as a consequence, the levels of acetylated histone 4 in the promoter is increased as corresponded with transcriptional stimulation of these microRNAs. Interesting, when we look both at the level of mRNA and at the level of protein to the expression of the putative target for the microRNA, we could observe that all of them were strongly repressed by T3. And in other experiments with T' UTR of this microRNA, we could observe that indeed T3 was able to destabilize the mRNA for these targets. But to really prove functionally that the inhibition of the expression of these targets by the microRNA was due to the increase by the thyroid hormone, we knocked down these microRNAs both in the presence and absence of T3. And interestingly, we could observe that this downregulation of the targets was inhibited when these microRNAs were knocked down. Then the increase in these microRNAs was probably mediating the reduction of these important targets for cell proliferation and mitosis, and also they are very important for the inhibitory effects of T3 in cell cycle progression. As you could see, the cells that are treated with T3 show increased percentage of cells in G1 with a concomitant decrease in the number of cells in the S phase. But when these microRNAs were knocked down, the inhibitory effect of T3 on cell cycle progression essentially disappeared. Therefore, this is an important component of the anti-proliferative mechanism of the thyroid hormone receptor beta in these cancer cells. But also another important role could the TR-beta to block proliferation could be to inhibit the response to growth factors. And not surprisingly, because these growth factors signal through RAS, we could observe that parental cells proliferated strongly in response to EGF or IGF-1, but the TR-beta expressing cells were unable to proliferate in response to these factors. But also very interesting, we could observe that the thyroid hormone receptor beta could abolish TGF-beta dependent proliferation. As you know, TGF-beta plays a very important role in the biology of cancer cells. This was also occurs in other cell types, for instance, neuroblastoma cells that proliferate in response to TGF-beta, and T3 is able to repress this TGF-beta dependent proliferation, and also to inhibit in wound healing assays the TGF-beta induced migration of the cells. We have analyzed, I will not go into this today, but we have analyzed the transcriptional mechanism by which the thyroid hormone receptor are able to repress TGF-beta dependent transcription, and we have seen that there is a direct interaction between the thyroid hormone receptors and the SMAD transcription factors that mediate the effect of TGF-beta and transcription, and this leads to repression of the SMAD dependent target genes. This is important because we have been able to demonstrate that the administration of thyroid hormone in vivo to mice is able to repress the formation of, for instance, liver fibrosis, that, as you know, is the first step leading to then cirrhosis and to the development of hepatocarcinomas. With respect to the inhibitory effects of TR-beta on metastasis formation, what we could demonstrate is that the thyroid hormone receptor is able to repress the transcription of many metastatic genes in a coordinated manner. And these genes are known to be of bad prognosis in patients, that had tumors expressing high levels of these genes. Among them, there are the chemokine receptors, the FCR4, and also other chemokine receptors, the proton cogenes MET, COX2, the transcription inhibitor ID1, and some metalloproteases. And in microarray analysis, we could observe that in the cells that were expressing high levels of TR-beta, the co-repressor NCOR was also upregulated. And then we reasoned that maybe that this repression of expression of this gene was due or was mediated by this increase in NCOR. And to prove this, we knocked down NCOR with siRNA, and we could observe that indeed this strong transcriptional repression by the thyroid hormone receptor beta was either totally or partially reversed in the absence of NCOR. Then these genes are more likely NCOR target genes, and this in chromatin immunoprecipitation assays we could observe that indeed NCOR binds strongly in the cells that express TR-beta, and this is accompanied by an increase in the recruitment of XDAC3, that is the main histone, the acetylase associated with NCOR complexes. Also, the promoter of these NCOR target genes had an increase of methylation of histone A3 in lysine 9, and as corresponding with the transcriptional repression, the histone acetylation of histone A3, the promoter was strongly reduced. And this, of course, was reversed when NCOR was knocked down in the cells. But NCOR is important, not only mediating the effect of TR-beta on the expression of the metastatic genes, but also in the cellular invasion, both in culture cells and in vivo. In invasion through matrigel, you can see that expression of the receptor reduces very significantly the invasive properties of the cells. But however, when NCOR is knocked down, this inhibitory effect is almost totally reversed. And the same happened when we conducted extravasation assays in vivo, and after a few hours after injection of the cells into the mice, we look at the cells that were able to extravasate from the blood vessel and to enter into the lungs. Expression of TR-beta reduces significantly cell extravasation, and when NCOR is knocked down, there is an important increase in the number of cells that can reach the lungs. Since the NCOR appears to modulate extravasation, we then look under these conditions to the formation of metastases after 1 month. And interestingly, we could observe that both the reduced incidence of metastases in mice injected with TR-beta expressing cells, as well as the number of metastases that we have seen before that were repressed by TR-beta, were, however, again reversed when the cells were knocked down. When NCOR was knocked down in the cells. The surprise was when we look at the expression of NCOR and the metastatic genes in the metastatic lesions that were excised by laser capture microdissection, and we could observe that 1 month after knockdown of NCOR with siRNA, it was not an shRNA, but with transient transfection of the siRNA, we could still observe an important reduction in the levels of NCOR transcripts, and also the repression of the NCOR target genes in the metastases. We then look at the tumor growth under the same conditions with parental TR-beta expressing cells in xenograft, and when the cells that were injected were transfected either with a controlled siRNA with the NCOR siRNA, I could observe that the tumors that do not express NCOR that have much higher growth. The volume is much higher, and also interesting this repressive effect of TR beta on tumor infiltration was reversed when the tumors, when the cells did not express NCOR. And here also, 1 month after injection, we could observe that NCOR was still knocked down and that the co-repressor target genes were upregulated. Then this indicates that there is a kind of autoregulatory mechanism by which, in the absence of NCOR, the NCOR gene is very stably silenced. And we studied this in the culture cells. You could see here that the experiments were conducted 70 days after siRNA transfection, when several doubling times had already occurred, and the siRNA should not be longer present. And even with longer time, we had similar results. You could see that the levels of NCOR transcripts and also the levels of protein of NCOR were totally depleted, and the NCOR target genes were re-expressed. And our first hypothesis was that since the NCOR gene contains CGP island, that maybe that promoted hypermethylation could be the responsible for the inhibition of the silencing of the NCOR gene. But however, we could not find changes in the promoter methylation at the CGP island of the NCOR promoter with after NCOR depletion. Then we look at other possible mechanisms, and what we could observe is that when we depleted NCOR, there was a strong recruitment to the NCOR promoter of the co-repressor or the co-repressor SMART or NCOR2 of the same family, and together with the recruitment of SMART, there was a strong recruitment also of the histone deacetylase 3, and this deacetylation of the promoter most likely is involved in this transcription and silencing, since when we incubated the NCOR depleted cells with the histone deacetylase inhibitor TSA, we could observe re-expression of the NCOR gene and reappearance of the NCOR protein in the cells. We look at other repressive marks in the NCOR promoter in the absence of NCOR, and indeed we could observe both in the parental and TR beta expressing cells a strong increase in the trimethylation of histone H3 in lysine 9. Also the polycomb dependent trimethylation of K27 in histone A3. And most interesting, we could observe that in the absence of NCOR, there is a strong appearances in the promoter of the heterochromatin protein H3.1 gamma. And this indicates that in the absence of NCOR, there is heterochromatinization of the NCOR promoter. And this heterochromatinization could lead to the depletion of transcription factors that bind to the promoter regions, and that could be important for transcriptional stimulation of the gene. And indeed, in the NCOR promoter fragment that we have been analyzing, there is the presence of 3 binding sites for the transcription factor SV1, that you know is very important for basal expression of many genes, not than the NCOR gene. And as you could see that under normal conditions, there is a strong recruitment of SP1 to the NCOR promoter, but when NCOR is knocked down, this transcription factor is displaced from the promoter. Then the model will be the following: that the NCOR has a very important role as a tumor suppressor and mediates effects of the thyroid hormone receptor beta invasion, stabilization, metastasis, and tumor growth. When NCOR is present at high levels in the cells. The NCOR gene is actively transcribed. SP1 is binding to the promoter, and this promoter is acetylated. But however, the opposite occurs with metastatic genes, in which NCOR is recruited through either the thyroid hormone receptor beta or other transcription factors, and then HDAC3 is recruited and the acetylation occurs, and also there's an increase in repressive marks. But when NCOR is not down, there is an increase in invasion, extravasation, metastasis, and tumor growth. The NCOR gene is silenced in a rather stable manner. This is heterochromatinization of the promoter with the acetylation and increase in transcriptional repressive marks. And however, metastatic gene expression can occur because HDAC3 is no longer present in the promoter. And we believe that this could be an important mechanism because it has been recently described that NCOR indeed acts as a tumor suppressor, and even in subtypes of tumors, including, for instance, in breast tumors, has been described as one of the driver genes in some of these breast cancer tumors. And I think that this autoregulatory loop that occurs with NCOR gene expression means that when NCOR, not only by mutation of the NCOR gene, but if NCOR is silenced for some reason, this leads to the loss of the tumor suppressor actions of this co-repressor and also of the thyroid hormone receptor beta. And to see whether or not we could find any of these changes in human tumors, we look at the expression of NCOR in normal liver, in the cirrhotic tissue that surrounds the tumor, and in hepatocarcinoma human tumors. And we could observe that most of the liver cells express NCOR, but however, NCOR expression is almost absent in hepatocarcinoma specimens. And at the level of NCOR transcript, both NCOR and TR-beta transcripts were very strongly reduced in human hepatocarcinoma tumors. And there was a very strong correlation between the levels of both transcripts, indicating that both genes could indeed act as tumor suppression in human hepatocarcinoma. But as you know, I'm sorry, some dissemination of metastatic formation in some type of tumors, and particularly in breast cancer, occurs mainly not through the blood vessels, but through the lymphatic vessels. The tumors contain lymph vessels that will transport the tumor cells to the sentinel lymph node, producing metastasis of the sentinel node, and from there they will go to distant places. And both in the development of normal lymphatic vessels and in the lymphatic vessels present in the tumors play a key role. Two factors, that is the vascular endothelial growth factor C and vascular endothelial growth factor D. And interestingly, in breast cancer cells, triple negative breast cancer cells that expressing thyroid hormone receptor beta, we could observe that the expression of the transcription of these genes was very strongly reduced with respect to the parental cells. And interestingly, we could observe that in the promoter of both vascular and bacterial growth factors, there are thyroid hormone, putative thyroid hormone response elements, and also SP1 binding site, and that NCOR binds very strongly to this promoter, and TR-beta also, mainly to the VGFT promoter, and that when NCOR and TR-beta is overexpressed, SP1 is gone from the promoter. And these indeed are NCOR target genes, as demonstrated by NCOR knockdown. You could see when we knock down NCOR in these breast cancer cells, both parental and expressing TR-beta, we could observe re-expression of this, or a large increase in the transcription of these genes. Then we look at the possibility that the TR-beta and NCOR could regulate lymphangiogenisis in breast tumor xenografts that were injected into the fat mammary part of the immunodeficient mice. And we look at the formation of the lymph vessels by expression of LIVE1, that is one specific marker for lymph vessels. We could observe that in the TR-beta expressing tumors, the formation of lymphatic vessels was very strongly reduced. But however, when we knock down NCOR, the vessels reappear. You see here that there's a strong increase in the number of lymphatic vessels. And most importantly, also the TR-beta and NCOR modulate sentinel node invasion. The number of cells that reach the sentinel node is very strongly reduced when the cells express TR-beta, but when we knock down NCOR, this is very strongly increased. And also in the cells, now not so surprisingly, we could observe that in the xenografts in the tumors 1 month after injection of the cells, NCOR was still significantly depleted, and however the expression of these prometastatic genes was strongly increased. To correlate this with possible effects in human tumors, we measure NCOR and TR-beta transcripts in comparing the levels in the ER estrogen receptor positive tumors that are much less aggressive and have a much better prognosis with the triple negative tumors that are ER negative. And we could observe that in the most aggressive tumor, there was a strong reduction of NCOR and TR-beta, and also there was a strong correlation between NCOR and TR-beta mRNA, both in ER and positive and ER negative tumors. And this correlation, in this case, negative correlation, we could also share between NCOR mRNA levels and mRNA levels of the LIVE1 and the lymphangiogenic factors, and the same happened with the TR-beta mRNA levels and the levels of transcripts of these lymphangiogenic factors, then suggesting that both TR-beta and NCOR would play a repressive role on lymphangiogenisis and probably also metastasis formation in human breast tumors. Then, in summary, we have seen that the thyroid hormone receptor beta inhibits drug-dependent proliferation, transformation, and tumorigenesis. This inhibition requires the co-repressor NCOR. That endogenous TRs act as suppressor of tumor progression in a model of skin carcinogenesis with mutation of RAS. That TR-beta induces cellular senescence. That, as I told you, the first barrier in cellular transformation and tumor progression. But we think that this induction of senescence may be also involved in well-known effects of the thyroidal status on longevity, and also in the occurrence of tissue damage that is associated to hyperthyroid conditions. We have also seen that expression of TR-beta reduces tumor growth, inducing transcription of microRNAs with tumor suppressor activity and inhibiting the response to growth factors and TGF-beta. That the receptor reduces tumor invasion and metastatic growth, repressing the expression of prometastatic genes and the transcriptogenic genes. And NCOR is an essential mediator of this repression. Also, that an autoregulatory loop maintains the expression of the NCOR gene, avoiding heterochromation of its promoter. And that TR-beta and NCOR expression are reduced, at least in some type of human tumors. And I think that this together really reinforces the idea that the thyroid hormone receptor beta can act as a tumor and metastasis suppressor and could be an important novel target in cancer. And finally, I want to thank you for your attention and the people at the Instituto de Investigaciones Biomédicas in my group, and our collaborators at the Universidad Autónoma de Madrid that conducted the histology, the people that generated originally the TR knockout mice, to Jesus Paramio and Tiamat, our collaborator Mario Fraga in Oviedo, Marisol Soemos de Cenio, and at the hospitals, people at the hospital, the Palacio de Aranda, that provided us with human samples, and al Ministerio de Carlos III and Comunidad de Madrid that funded our research. And finally, I want to thank these children that they make my life much more fun than it was before. Thank you very much.
Hi.
Hi.
Thank you, Anna. Very, very nice. So there's several questions that the audience was asking. I'm gonna try to compile them because there were so for several people have also similar questions. So one of the questions that several people have asked is if I've seen some results with TSA, but if you have tested further the effects of HDAC inhibitors and if they are similar to NCOR.
No, we haven't, but most likely other HDAC inhibitors could have an effect. Really, I mean, one of the effects of HDAC inhibitors as suppressors of tumor growth could be one of the targets, could be NCOR.
Mm-hmm. Okay.
No one has tested it still.
Mm-hmm. I mean, you have shown that it's HDAC3, no? The one that is interacting with the NCOR here.
Yes, because in the NCOR 1 or 2 complexes, it has been demonstrated that the main HDAC is HDAC3.
Mm-hmm.
By the way, it's altered in hepatocarcinoma models and in vivo also in humans.
Because in relation to a potential antagonist effect, no, like so you at the beginning of your lecture, you said that the thyroid hormone receptor also interacts with coactivators and histone acetyl. So I mean that pathway would be a potential druggable pathway, not to have a opposite effect, no?
Yes, probably. Yeah, I mean that what we haven't tried the effects of some of the knockdown, for instance, of coactivators in the expression of some of the genes that could most likely be recruited to when TR beta is present. But this is transcriptional inhibition that is like you know, it's not the normal mechanism that one thinks when one is thinking in transcription factors such as the thyroid hormone receptor, because one would think that transcriptional stimulation would be very important. But I think that our data, they really say is that transcriptional repression could be more important than transcriptional stimulation in the antitumorogenic effects and of the thyroid hormone receptor, not in other effects that depends on the classical T3 dependent transcriptional activation.
A little bit also related, and I mean, of course, there's a lot of interest here in relation to these histone related effects. So some of the attendees was mentioning that sirtuins, and particularly SIRT1, has been associated also to TR beta function. So if you had or if you knew of some results in relation to the sirtuin mediated effect of this TR effect in cancer?
In metabolism, there are some studies, but not in cancer, really. I mean, we are analyzing we know that, for instance, in immortalized immune cells, T3 is able to induce sirtuin, the levels of protein itself, not the activity, but the levels, also the levels of protein. But no one has still studied the effect of the potential effect of thyroid hormones on tumor cell. Mediated by sirtuin, but most likely, since the thyroid hormones such a strong metabolic effect, I'm sure that some of them are mediated by sirtuins. And I mean, we don't have direct data. What I could tell you is that when we, for example, when we conducted experiment of xenograft formation in hypothyroid hosts, nude mice, we could say something interesting is that the tumors are smaller, and we thought that it was due to the metabolic effects of thyroid hormones, probably in the surrounding cells, because the tumors were also smaller when the cells that the tumor cells did not express the thyroid hormone receptors. I mean, it's very indirect, but we are almost sure that the metabolic effect of thyroid hormone could be involved in the antitumorigenic effects and in many other effects of the thyroid hormone receptors.
A little bit in line with also the oxidative stress that if it seems to be important for the TR anti-tumorigenic role. So would perhaps administration of antioxidants or could have a compensatory effect, or do you know?
It has in the culture surface. It does have an inhibitory effect. I mean, the effects of T3, the increased premature senescence induced by the hormone is reversed if we treat the cells with antioxidants. So we haven't tried in vivo, but most likely, well, it's interesting that when in a strong hyperthyroidism has been suggested that the treatment with antioxidants could be a good way of controlling some of the what they call the thyroid hormone storm that occurs in very strong hyperthyroid conditions. That everything fits with the idea that antioxidants could be important inhibitors of these deleterious effects of the thyroid hormones when it's a large excess of thyroid hormones circulating.
Okay. Also, in relation to the regulatory, the sort of genomic effects of the TR, so, and also epigenetics. So there's I mean, the past few years, like a lot of interest on enhancers and other regions. So I don't know if you have perform or other collaborators work on with ChIP-seq or even just the presence of the TRE is in enhancers and instead of promoters.
There are some studies. It's incredible, but the antibodies for the thyroid hormone receptors are not very good. But despite this, there have been some studies mainly in the liver, and there are indeed thyroid hormone binding to enhancer regions, and it's very similar to what happened to other nuclear receptors. I mean, they bind preferentially to enhancer regions. They bind everywhere at the genome, and the promoter regions are a small percentage of the total binding TR binding sites in the genome.
No, so I guess that when eventually there are good antibodies, so there could be a whole world, in relation to long distance effects and effects on structure.
I mean, there are not as many studies with the thyroid hormone receptor, for instance, the estrogen receptor or other receptors that have been much more profoundly studied in this type of 3T or 4T and all these enhancers, and much more than the thyroid hormone receptor. Not many studies on the thyroid hormone receptors, because not even with ChIP-seq, not many of them have been performed.
Okay, and just one final question. So there were some people asking if you had explored more on human cancers, and if you had a sort of feeling of which type of cancer might be sort of a main target for this thyroid receptor mediated pathway.
What has been described is that, for instance, there have been found many mutations in clear renal cell carcinoma, mutations of the TR-beta, and also something interesting is that in this type of tumors there is a kind of local hypothyroidism, because the metabolism of the thyroid hormones, I mean the formation of the active hormones T3 is reduced in the tumors, and also in hepatocarcinoma appear to be very good target for TR-beta. I mean, this has been demonstrated that in early almost pre-neoplastic lesions in experimental models in the rat, TR-beta plays a very important role. I mean, this probably hepatocarcinoma is one of the tumors in which the thyroid hormone receptor beta is more important. It's interesting that all these tumor suppressor effects of the TR-beta are not shared by TR-alpha. For instance, in hepatocarcinomas, this has been described that TR-alpha is high instead of low. It's just the opposite of what happens with TR-beta. And so we haven't studied it, but there is the idea that TR-alpha, TR-beta could have opposite effect, that would be a kind of yin-yang. Almost a little bit like it happens with ER-alpha and ER-beta.
Okay. Well, I think, well, thank you very much for presenting this exciting talk, and we hope we can host you soon here.
Okay.
Okay. Thank you. Thank you very much, and thanks everyone. Bye.
Bye.